everyone. Our group includes Chantal, Nick, Alyssa, and Ronia. Have you ever wondered how a heavy metal inhibitor will affect the reaction between the enzyme catalase that is found in the liver and hydrogen peroxide? Will it slow down the reaction or speed the reaction up? Well, that is why we are here to tell you what happens. Our hypothesis is that if a heavy metal inhibitor is added to a liver sample with hydrogen peroxide, then the enzyme reaction will speed up. We thought this because we knew that it was a copper inhibitor, and we assumed that it might act like a cofactor. Let's find out if our hypothesis is right or wrong.
To conduct this lab, we use the following materials: 2 1-gram samples of liver, 20 milliliters of 3% H2O2, a heavy metal inhibitor which was copper sulfate, 3 graduated cylinders, an electronic balance, 1 beaker, 1 test tube, a weighing boat, 2 stirring rods, a stopwatch, and gloves, aprons, and goggles.
After we gathered some of the materials, here's how we conducted our experiment. First, Alyssa measured 1 gram of liver into a weighing board using an electronic balance and placed it into a graduated cylinder. She then did the same thing and placed 1 gram of liver into another graduated cylinder. We then used a small beaker to get the heavy metal inhibitor, which is copper sulfate. In order for the inhibitor to work, it had to sit with the liver sample for at least 5 minutes. So Alyssa poured about 5 ml of the inhibitor into one of the graduated cylinders, just enough to cover the liver. We then let it sit for about 20 minutes. While that was sitting, we measured 20 ml of 3% of hydrogen peroxide into another graduated cylinder. We then divided the 20 ml in half by pouring 10 ml into a test tube. Once the 20 ml was divided, and after the 20 minutes, Alyssa poured 10 ml into one graduated cylinder, while Chantal poured the other 10 ml of hydrogen peroxide into another graduated cylinder at the same time.
As you can see in the video, the control reaction that had no heavy metal inhibitor had a much greater reaction occur. Within seconds of pouring the H2O2 into the graduated cylinder, bubbles and air rose to the top and filled the graduated cylinder. This is due to the enzyme catalase in the liver reacting with the hydrogen peroxide. The catalase broke the hydrogen peroxide down into water and oxygen, which is why the graduated cylinder filled with bubbles so fast. As you can see by the video, the graduated cylinder that had the heavy metal inhibitor in it did not react. There was no reaction due to the heavy metal inhibitor, copper sulfate. The presence of the heavy metal inhibitor caused the catalase enzyme to denature. The shape of the enzyme was destroyed. Due to this change in shape, the hydrogen peroxide, which was a substrate, was not able to bind onto the active site of the catalase, so the hydrogen peroxide was not broken down to release the products water and oxygen, like how it was in the controlled test.
Our hypothesis was not correct. We assumed that copper would act like a cofactor and help speed up the reaction due to oxidation, but it did the opposite. It inhibited the reaction.
Potential sources of error in our results include the 2 liver samples may not have been exactly equal, because when we measured one, it was 1.03 grams, and when we measured the other one, it was more than 1.03 grams. But we did try to remove or add some liver to make the amounts equal. Another source of error is that when we divided the hydrogen peroxide, it might not have been exactly divided into 2 10 milliliter quantities.